Section 6.3 deals with enzymes and metabolic pathways. Metabolic pathways are a series of linked reactions that happen in living organisms. And these begin with a specific reactant and produce a specific end product. And the reactant of one is the product of the next, and that becomes the reactant of the next pathway. Enzymes are proteins that function to speed a chemical reaction. It's really, really important that you remember the four categories of macromolecules in living systems, and they are the carbohydrates, the lipids, the proteins, and the nucleic acids. It's really important that you remember that proteins, one of the proteins are enzymes, so enzymes fall under the protein category. And these function to speed up a chemical reaction, which means that they are a catalyst. In chemistry, a catalyst is something that speeds up a chemical reaction, but is not involved directly in the reaction. So it isn't part of the reactant and product. It starts out as a reactant, ends as the same product. Here is an example of a metabolic pathway. So we have reactant A is going to be helped by enzyme 1 to form product B. Product B is going to become the reactant of the next reaction that's going to be sped up by enzyme 2 to form product C, which is the reactant of the next reaction, and so on. So a metabolic pathway is one that has the product of one becoming the reactant of the next chemical reaction that needs to occur, thus making chemistry the central science. Because even in biology, without the chemistry going on, you don't have a living organism. These reactants are also called the substrates. Energy of activation, we talked about this in thermodynamics in chapter 10 in chemistry 2. The energy of activation is the energy that must be added to cause molecules to react with one another. An example of this would be needing a match to start wood burning. The wood is going to spontaneously combust. Something has to cause the reaction to start. Enzymes lower the activation energy that's required for a reaction to occur. They don't change the end result of the reaction, they just speed it up. So here's an example. If you look at the blue color, the blue is the reaction when the enzyme is not present. So the activation energy required in order to take the reactant to product is quite high. So it's this blue bar right here. However, if I put the enzyme in the reaction, look what happens to the energy of activation, how it's lowered. So it takes less energy to go from reactant to product with the presence of an enzyme. So how do enzymes function? Enzymes bind a substrate to form a complex. So an enzyme plus a substrate is going to form an enzyme-substrate complex, and that's going to release itself, and you're going to have an enzyme and a product at the end of the reaction. So let's look at the difference between an enzyme and a substrate. Enzymes have a place called an active site, and this active site is where the substrate is going to come in contact with it, and it's going to allow it to speed up the reaction that's going to take place. In this example, it's a degradation. In degradation, the substrate, which is this, is going to be broken down into smaller products. And you see that that's what happens. The substrate is going to come in contact with the active site of the enzyme. The enzyme is going to speed up the reaction. The reaction in this case is taking this substrate into two smaller substances. The substrate binds the active site on the enzyme substrate complex, then you have the enzyme released and the products are released. So the substrate's gone from this to its two things that make it up, or the two components of it, and it could have done it on its own, but the enzyme sped up the reaction. Synthesis is making, so the enzyme can also have an active site when the two substrates in this case, instead of one, go in here. It's going to speed up the chemical reaction to join that chemical bond and form a product. And that product was formed by putting these two substrates together. There's a model called the induced fit model. There's a model called the lock and key model. The induced fit is the one that's being a little bit more widely accepted right now. And this says that the substrate and the active site shapes don't match exactly, but the active site is induced to undergo a little bit of a change in its shape to accommodate the substrate that's binding. And this change in shape helps the reaction to occur easier. So in this case, you notice that there's this active site here, and the substrate kind of goes in there, and it gets surrounded, it mushes in there kind of like with Play-Doh. So the active site is induced to fit the substrate. and that's the model that's been accepted more recently. Reactions require a specific enzyme, and enzymes are easy to identify because you're going to notice that they always end in ASE. For example, for a lipid, the enzyme that is going to be used as the enzyme for the substrate is lipase. For urea, it's urease. For maltose, it's maltase. Remember that OSC is indicative of a sugar. ACE is an enzyme. And the enzyme lactase is going to break down the sugar lactose. Factors affecting enzymatic reactions. So substrate concentration is one of them. So the more substrate that you have, the more likely the reaction is going to occur at a faster rate. Temperature and pH, enzyme activation, and enzyme inhibition, as well as the cofactors. So let's take a look at each one of those. Substrate concentration. Enzyme activity increases as substrate concentration increases. This should make sense because the more substrate you have, the more collisions there are going to be between the substrate and the enzyme. That's going to speed up the reaction. So the maximum rate is achieved when all the active sites of the enzyme are filled continuously with the substrate. The substrate hooks up with the enzyme, the reaction occurs, the substrate is released, a new substrate comes in. Temperature. As you know, temperature is going to speed up activity. So enzymatic activity increases as temperature rises. So the higher the temperature is, the more effective the collisions. However, you have to be careful because enzymes are proteins, and proteins at high temperatures can denature, which makes them lose their structure. It unwinds them from their secondary, tertiary structures, and it causes the function to be gone as well. So although the temperature is an increased temperature is going to increase the activity, too high of a temperature can cause the enzyme to break down. pH. Each enzyme has an optimal pH level. Enzyme structure is pH dependent, and remember pH is whether something's acidic or basic. Extremes of pH can cause denaturation, which is what happens with temperature extremes, and it can alter the enzyme structure. This diagram just shows optimal levels for different enzymes. Pepsin is an enzyme, and so is trypsin. Pepsin works best at a pH of about a 2. Now, it will work at a 0, but just not as effectively, and at a 4, but just not as effectively. Trypsin, on the other hand, doesn't work at lower pHs. It works at more basic pHs. So its optimal level of working is around a pH of an 8. Enzyme activation. Cell regulates metabolism by regulating which enzymes are active and which ones are not. We can't have them all active at the same time. Genes producing enzymes can be turned on or off to regulate enzyme concentration. And enzymes can be modified by adding or removing a phosphate, which changes the shape of the enzyme. Enzyme inhibition occurs when enzymes cannot bind to its substrate, so it's not allowed to. The activity of almost every cell enzyme is regulated by feedback inhibition. When the product is abundant, so there's a lot of it there, it binds to the enzyme's active site and it blocks further production. When the product's used up, it's removed from the active site. The enzyme begins to function again. For enzyme inhibition in more complex types of inhibition, the product binds to a site other than the active site, which changes the shape of the active site. Poisons are often enzyme inhibitors. Cyanide inhibits an essential enzyme, and penicillin blocks the active site on bacterial enzymes. Let's take a look at an active pathway. So the first reactant A fits into this enzyme E1, and this is the site where the end product F can bind, right here. This is an inactive pathway where this is altered. This where A should bind is altered because there's so much end product that the end product will stop the ability of the first reactant to fit into the site on the enzyme. So the site of the enzyme where F binds, and we have so much of F built up that it actually will go into this site here and change the shape so that A can no longer fit into the enzyme to continue this pathway. Enzyme cofactors. Molecules that help the enzyme function are called the cofactors. Copper and zinc are examples of inorganic cofactors. Organic non-protein cofactors are called coenzymes. And co means to do some, like co-ed is boys and girls together, so co means together. Cofactors and coenzymes work with enzymes in order to help the function of the enzyme. Vitamins are often components of coenzymes as well. So this ends the section on enzymes. It's a very important section. Make sure you take time to take your notes and pause the video as you need to in order to get your notes filled in.